Okay, so lab 1 is we're going to go over the value of controls. And so that's summarized in page 6 of your lab manual, which is the first page of the packet that I have for you guys with the little smiley faces on it. We just did the lab Ubiquity of microorganisms, and so we're going to talk about that. We're going to introduce the light microscope, and next week you will actually use the microscopes that you picked your numbers for. So some important definitions. Sometimes when you run experiments, you're going to end up with what's referred to as a false positive result. And so I always use testing that most of us are familiar with as examples for this. So what's the chance that you run a pregnancy test and it comes out positive but you're not actually pregnant? Very rare. Very rare. Right? And we're going to talk about why that is. So that would be an example of a false positive, right? Where the test says you're pregnant, but you in fact are not, right? So that means you observe a positive result, right, for your sample, but you expected it to be negative, right? You're like, I'm not pregnant. A false negative. What does that mean in terms of a pregnancy test? All right, you are in fact pregnant, right? But the test says you're negative. So you observe a negative result, but you expected to see a positive. How could this happen? Well, because tests work within particular ranges of abilities. One of them is referred to as sensitivity. This is a limit, right? You have to be within that range for it to say positive or negative. So this is typically where you get a false negative result, right? So you're pregnant, right? But the pregnancy test says negative, right? But hey, you've been trying, right? You know you haven't had your period. You're like, I gotta be pregnant. Why is it not saying I'm pregnant, right? What's going on here? What's going on is that you're testing what when you do the pregnancy test? You're testing, she's got it, human chorionic growth hormone. But where are you what type of sample are you testing? A urine sample. That hormone, right, only ends up in your urine when you produce a lot of it, right, and that excess gets taken out of your blood and ends up in your urine. So where is going to be a higher concentration? In your blood and in the morning, right? So we're dealing with sensitivity. You don't have enough of that hormone probably in your urine for it to be detectable by that test. Now these tests have become very sensitive, right? They tell you can test, I can't even remember how many days after your first missed period. I mean, they're getting really sensitive, right? Where really small amounts of that hormone can be detected. So when you go to the doctor's office, right, and you've done the urine test, it's negative, right, but you're pretty sure you're positive, they're going to take a blood sample, right, because that's going to have a higher concentration of the human chorionic growth hormone that you only produce when you're pregnant, right, or unless you're dealing with some abnormality, right, which can happen too. Right, yeah, I mean, that's what I'm saying. There are other things, but, you know. So, specificity, right? How specific is this test? Well, it's pretty darn specific, which means that if it comes out positive, don't do like Juno did in the movie and take like 3 or 5 more different pregnancy tests, because what did they do? They all came up positive, right? You can't make it go away, right? Once positive, positive. Go figure out what you're going to do about it. I was excited, right? I took a picture of mine when it came out positive. I wanted to be pregnant. I wanted to be a mommy. But this is a pretty specific test. So if it's positive, don't bother. It's positive, right? Or go figure out what else is wrong. So I don't know how this happened. Maybe some guy had sympathy for his wife who had to pee in a cup. But women, men can also take this test. And apparently, it will react with some other substance that's overproduced when men have, I can never say this word, testicular cancer, right? So they're actually using it now as an early detection for that. Pregnancy tests. Who knew? Does that make sense to you guys? Right? So if it's a false positive, right, it's usually a specificity issue where something else, right, it's not a very specific test. It can react with other things. So in the case of the pregnancy test, right, it can react with other things, but not other things that women produce. It's other things that men produce. Specificity comes into play a lot with drug testing too, right? Where certain drugs, right, can cause other drug tests for other substances to come up negative. Why was this going off again? So I'm going to ask Aaron, I'm going to get it eventually, to label this for us. And so we're going to write 30 minutes air, right, date, class, and what temperature, y'all? Yeah, let's do room temperature. So controls are done to help us understand our results and have a level of accuracy to them. Yeah, you can go ahead and put it in there. We are doing the, you hit the button. Flip it over. So when you're running tests where you get a yes or no answer, that's a qualitative test, right? A pregnancy test is a qualitative, right? Yes, you are. No, you're not. So the pregnancy test has a built-in positive control, because that's what we care about, right? Is the test working properly? Can we get a positive result? And so if some of them have those digital readouts and stuff now, but if you take that off, you'll see two lines if you're pregnant. One is the positive control, and one is your test sample that came out positive. Two lines is pregnant, positive, right? One line means the test worked, right, but your sample came out negative. If you have no lines, does that mean you're not pregnant? No, what does it mean? The test didn't work, right? And usually it'll say error, repeat, right? So maybe it was expired, who knows, it just didn't work, right? So you got to do it again. So in the case of our experiment today, we have what type of control? Negative, right? Because we want to make sure that our media when we started was what? Sterile, right? We don't want any growth. If we get any growth, that's going to mess up our experiment because we don't know, is it contamination on that plate or did it come from someone's fingers or from the mouth sample we did or the air sample? We don't know, right? So if we were in a real lab, what would we have to do? Start all over again. Are we going to do that? No, this is a teaching lab. Right? We learn from when things go wrong. Okay. Standards are one step higher, you could say, from controls. And these help us when we want to do quantification, if we want to get a number result, right? We don't want to know that you're positive. We want to know how positive you are, right? So in the case of your antibodies, those of you guys that are getting ready to go into nursing school, you're going to have what's referred to as a titer check on all your immunizations. They want to know how many antibodies you have against the things you should be vaccinated against, right? How much immunity do you have, right? And they're going to actually get a number for this, right? They're going to figure out about how many antibodies you have against things like measles, mumps, rubella, right? In order to do that, you have to have a control that will help you determine this. So you don't want just a positive control. You want a control that says, we have 0.5 antibodies. We have 1 microgram per milliliter of antibodies, right? We need a range of positive controls. This is referred to as a standard or a standard curve. Typically how they measure these things, they use a machine called the spectrophotometer. We have one. Hopefully later in the semester we'll actually do an assay where we're looking at antibodies and we're actually going to figure out the number of antibodies, the titer, the concentration. In order to do that, we'll have to set up a standard curve. So we're going to need known samples. Your unknown, of course, is something you don't know the value of, right? And we would have to compare that to our known values to figure out what it is. So here's an example of a protein assay. So they have set numbers, right, concentrations of the protein, and then they have, so this is a set concentration, right, 4 micrograms, 8, 12, 16, 20. And then they read it on the spectrophotometer to get a number associated with that, right, the absorbance. So these are those numbers. So when we graph that, and you could do a straight line graph or you can make a graph conform to all those dots, right, which would be more accurate. But it depends on, how accurate you need to be. And you can do the math associated with it. The good news is nowadays you can plug these into computer programs and it does it for you, right? I actually didn't go to school that long ago that I had to do it by hand either. I did it on the computer. But you could take your unknown sample. So say your unknown sample and you read it on the spectrophotometer and you get a number that lands here. And you base that on the standard, you could figure out that the concentration is, a little bit less than 10. And you can mathematically calculate this, right, using a computer program or the formulas seen here. So we can actually get a real number, right? Not just, yes, the protein's there. And we can say, no, there's about, say, here, where is it? 77.1 micrograms, right? Based on that absorbance by the spectrophotometer. So hopefully towards the end of the semester we'll actually do an experiment like that where you guys, instead of just positive, we're going to get a number. We're going to know how positive the sample is. And for sometimes you need that level of accuracy, right? You want to know how much is there, not just that it's there. So we've already kind of talked about these, but they got cute little smiley faces, right? And mathematical formulas to help you understand, for those of you guys that are math-minded. All right, the importance of having controls and having false positive and false negative results. So when we deal with false positives, right, in our control set, right, it's probably a specificity issue, right? Again, what you're testing for, what you think you're testing for, something else is causing it to be positive, right? And there's one test that we do run. If you don't run it properly, you don't know if you're testing for that enzyme or the metal loop that we use is causing the reaction. It's called the catalase test, right? That enzyme can be turned on by your organism or it can be turned on by metal, right? So you have to perform that test properly so that you don't have a specificity issue. You don't get a false positive result, right? Because false positives are going to be misleading, right? And especially if you're trying to figure out which unknown organism I gave you, you're not going to get it right if your results are not accurate. So you want to be accurate in your results. If you get a false negative, right? It looks negative, but your organism should be positive. This can be a sensitivity issue. Again, for the tests that we run, we know the ranges of sensitivity, right? So we have an idea if there's a possibility, right, that something went wrong. But it's also why we run controls, right? So that we get the positive and negative results we should get. And you have a comparison. You know the test was run properly. Right? So assuming you haven't had any catastrophic errors in technique or protocol, right, you should get, right, your positives should come out positive, your negatives should come out negatives for your controls. If they don't, then you're dealing with sensitivity or specificity issues. And of course, we're going to be using this throughout the semester, right? So if you don't quite get it now, this is terminology we're going to keep using over and over again. So these, unfortunately, we have reported this to the publisher, but it didn't get fixed in the last go-around. They fixed the equal sign. It used to be a 4 there in the lab manual. But one of these equations is wrong. I think it's the specificity one. Remember, we're looking for false negatives, right? Those are what affect our specificity. If we don't have any false negatives, then we're not dealing with a sensitivity issue. For false positives, it's a specificity issue. So hopefully you have 0 for both of these, and then you don't have sensitivity or specificity issues. If you have any false positives or any false negatives, right, there's some issues with your test. So we've already mentioned ubiquity. Ubiquity means everywhere, right? So microorganisms in general can be found everywhere. Not every microorganism is in the air or in our bodies, right? But in general, can you go anywhere on this earth or this room and avoid microorganisms? No, it's not going to happen. They're everywhere. They're ubiquitous. What's a pathogen? It's a microorganism that will, right, we know it causes disease. We know it could make you sick. Something that's non-pathogenic. Right, but this is the important part of that one. It's not known to cause disease, right? So we could say something is non-pathogenic, and next week some scientist discovers it does cause some disease. So it doesn't mean you're 100% safe if we say non-pathogenic. Just means most likely it's not going to make you sick, to the best of our knowledge. Opportunistic pathogen. So what does opportunity mean? Availability, right? The conditions have to be right for this organism to make you sick. A lot of your gut microbes, what we commonly and generally refer to as enteric bacteria, right? These are the an informal group of organisms that inhabit the gut of warm-blooded mammals like ourselves. So E. coli is the big number one that most people are familiar with, right? That's fine if it's in your poop, right? If it's in your intestinal tract, it's not going to make you sick. The problem is, little girls are taught, when we're very young, a particular way to wipe after you've gone number 2, right? You got to wipe to the back. Why? If you go forward, what's going to happen? You're going to get poop in your vagina or urethra. And do you want that there? No, because what's in that poop? E. coli bacteria, right? If they get into the vagina or the urethra, they can cause serious problems. A little girl in Louisiana, I kid you not, died from something as simple as not wiping properly. Got an infection of her urethra, right, to the bladder, up the ureters to the kidneys, damaged her kidneys and killed her. It's not a laughing matter. And guys, don't tune me out either, Andrew. Who knows, you might have a little girl someday, right? So you've got to take care of, right? This is important stuff. Guys don't have that problem, right? They only have 2 holes and they're pretty far separated. Of course, it varies, but still, you know, you really have to work at it to get it to the urethra. Then we have other issues. So then I'll tell you the story about one of my other male students who was babysitting his little boy and was not paying attention to him. And came back into the room and the little boy had poop everywhere and was eating it. Yeah. He didn't get sick. He's like, Erica, I don't understand. I said, Erica, I don't understand. How come he didn't get sick? I said, because it was his poop. He just put it back in where it came from. Isn't that a problem? It's when you get someone else's poop that might have something in it that doesn't agree with you that you can get problems. But animals do it all the time. Dogs will eat their own poop because it still smells like food. Horses, cattle do it on purpose because the babies eat their mom's poop to establish their microbe environment, right? You get your microbes from your mom, from your environment. You establish your normal gut microbes. And actually, some of the healthiest people out there, the in the world of the Indian tribes in Africa that roll around in the mud and are we would consider them dirty, but they're some of the most healthiest people because they have a very diverse microbial population in their body. So you can actually be too clean, right? This is why we all have to take probiotics and stuff, right? Because we become super clean, crazy people, right? It's okay. I ate dirt pies when I was a kid, right? With Jimmy's, I could eat the ants back home. They weren't fire ants. Host. What do we mean when we're talking about host? So what are you when you're a host? Everyone came to your house, right? So in this case, you are the house for the microorganism. If you're a host, you are the house, right? You are the habitat for another organism. This relationship could be bad, could be parasitic, or it can be commusal, which is, that relationship we have with our gut microbes, right? Those E. coli that do good stuff for us, like make vitamins for us, right? And help us digest our food properly. So commusal means, right, that it's a synergistic relationship. We're both acquiring the same home. But neither one benefits or is negatively affected. So this is communism, right? Everyone gets a share, an equal share. Nobody is negatively affected. No, I don't believe that that's possible, and I don't believe that's truly possible for microorganisms, right? Somebody is benefiting to a certain extent. You might not be negatively affected, but somebody's always getting something better than someone else. Mutualistic is all about the positive, right? Both organisms benefit from the relationship. So definitely our E. coli, they benefit. They have a place to live and food to eat. And they make vitamins for us. That's good for us, right? That's a win-win situation. And how I always remember mutualistic is how a good marriage really should be, right? Marriage should be mutualistic. Both partners should benefit. If you don't, then, you typically get divorced. What's a reservoir? So this typically is non-human, right? Although we are animals, we put ourselves on this little pedestal, right? We take ourselves out of the animal equation, keep ourselves separate. So animals other than humans, or other sites in nature that serve as a perpetual source of pathogens. So reservoirs are typically things we want to avoid, right? So in Louisiana, what's the number one reservoir? What's the number one animal you want to stay away from in regard to rabies? Raccoons. Raccoons are number one. Leave those cute little guys away. Leave them alone. They're nocturnal. If they're out during the day, that's not normal behavior. If they're coming up to you, that's not normal behavior. They should be running from you. Leave them alone. At the vet school when I worked there, when people bring them there, they kill them and autopsy them to find out if they had rabies. 98% of the raccoons dropped off there have rabies, even as babies. Yeah. Okay. Oh yeah. It's bad news. So it's not cute. It's not cute. No. Shoot it. Shoot it and call animal control. Yeah, well, raccoons can eat your cats. Let's say you're feeding the neighbor's cats. Yeah. I mean, they'll come eat cat food, yeah. Right, so there's a lot that's going to hurt the cats and they're going to get rabies. Yep. Yep, not good. Not good. Yeah, definitely, I mean, if you feed feral cats, feed them in the morning when the raccoons aren't going to be around, and only feed them enough that they're going to eat it right away. So we have a lady that comes into my neighborhood and feeds the feral cats. She puts the food on the sidewalk. The kids come out in the morning. The kittens, the kids, fur babies. They come out and they eat the food, right? She only gives them enough that they eat it all up and it's gone. So there's no other animals coming along and eating it and contaminating their food. Although I don't think we have any raccoons in the city. I hope not anyways. Well, yeah, that's true. We've got City Park. Darn. Yeah. So, yeah. I mean, I know I had a possum in my yard at one time, too. So, free living, they don't reside on or in a specific host, right? And typically, these are non-pathogenic organisms when we say they're free living. Because they're not required, right? So what's the purpose of our unopened plates? What's a label that we put on them? Negative control, right? They show sterility, right, of our media, the food that we have there for the microorganisms to eat. So our appropriate label is negative control or no exposure. What else do we label on our plates? What temperature we're going to store it at? I'm waiting for you guys. What do we put on those plates? We put the temperature, we put the date, the class. I think that's all. Date, class, or name, right? Temperature. Where do we label the plates? On the bottom, where the food is. Otherwise, someone could switch the lids on us and really mess us up. Same is true with our tubes. We label the tubes, not the caps. The caps could be switched. The lids could be switched. Typically, we write along the edge, right, so that you can better visualize the growth on the plate. I didn't have you guys do that today. It's not a big deal. What happens if we have growth on those plates? Our media was contaminated, right? So that media, the food source, was probably contaminated. If you were in a clinical or research lab, what would you have to do? We'd have to start over, right? We'd have to repeat the experiment because we would not know the source of the growth on those plates from contamination or what we were testing. What if growth only appears on one of the plates? Does this affect the reliability of our experiment? Yeah, so say the 37 plates got growth on it. That means we don't know for all our 37 plates, but it puts our whole experiment into question, right? So how is it, though, that only one of the plates, the 37, grew and not the 25? Maybe the contaminant. Does temperature affect growth? Yeah, so maybe the contaminant only grows at 37. Does that mean it's not on our 25 plates? No, it could be there. If we stuck those at 25, it might start growing. Right? It could mean, the contaminant is temperature sensitive. It could mean maybe that only a single plate got open. Like when I was over there, right, I noticed one of them was open. I put it off to the side. Right? We don't want to use that plate. It's potentially been contaminated. Does that make sense to you guys? Okay. So any growth cast doubt, right, on our experiment. So these are some pictures of some previous ones, right? You can notice that the 25 versus the 37 for the desktop, notice the differences in growth, right? And this is the same source, right? So the difference here is based on what? Temperature, right? And how organisms are affected by the different levels of temperature. Here's the whole class fingers. Look at that. And that was only at 25 degrees. Look at how much growth is on that plate. Look at the soil one. Look at our air one. And look at this fuzzy stuff. Is that bacteria? It's fungi. It's mold, right? It's mold. It's going to be fuzzy like that. So that's typically why I have you guys do the soil of the leaf sample, because up until that one crapped out, we had a HEPA filter running in here, which eliminated a lot of the mold spores in the room, which helped keep our stuff from getting contaminated with mold. But it's in the air, or you brought it in. So these exposures were chosen because, again, to represent the fact that microorganisms are everywhere, right? We tested the air, the soil, our fingers, the desktop, our nose, our mouth. Microorganisms are found everywhere. So our two different temperatures in this lab represent what? They represent room temperature for the 25, which would be around 77 degrees Fahrenheit. I would actually prefer it to be cooler than that. 70 would make me happy. 37 represents 98.6, which is body temperature. That's why we utilize that. The Celsius scale is based on the properties of water. It freezes at 0 degrees Celsius, which is 32 degrees Fahrenheit, and it boils at 100 degrees Celsius, which is 212 degrees Fahrenheit. 30 would represent soil or skin temperature, right? It's about 86 Fahrenheit. What's the likely source of the organisms that grow best at 37? Us, people, right? How do you think they survive at room temperatures without nutrients? So on that desktop, are we going to pick up ones from us? Yeah. Are they going to grow at 37? Yeah, because we gave them a whole lot of food and we made it nice and warm and happy. Okay. Are they growing on that desktop? They could, right? Because even though you don't see them, they're there, right? So some organisms grow better at room temperature, where others, especially ones that come from us, are going to like 37. Most of them that grow at body temperature will also grow at room temperature, but not as fast as they would at 37, especially once you give them food. Hence why that fingertip one, right? Lots of growth, even though it was at a cooler temperature. It's also because there's lots of organisms and lots of food. Right? We put them on food. Microorganisms are also very good. Right? There's maybe a small amount of nutrients on those desktops, but they're also very good at starving. They're very good at going without for long periods of time. Much better than we are. So, one, there may be nutrients there. Two, they're really good at not eating, just chilling until food becomes available. Some of them even can go into long stage resting forms known as endospores as far as bacteria goes. Right? So things like anthrax and tetanus and botulism, all those bacteria can cause the formation of endospores. These are highly resistant to environmental conditions. You can't boil them to kill them. You have to incinerate them, inflame, like we do in lab, right? Or autoclave them, how we sterilize and destroy our microbes before disposing of our stuff. You can't dry them out. You can't, most of them are still resistant to radiation. Most chemicals can't penetrate that spore coat to destroy the DNA, to kill them. So that anthrax scare, that powder, was millions upon billions of spores. Once those enter into somebody's body, they germinate. They form back the original cell they once were. And the bad news about that bacteria, right, it will destroy your body and kill you. Even with treatment, depending on the level of exposure, someone could die from something like anthrax. This is why it's pretty scary for us. The good news is in the United States is that most of the instances of it, we've cleaned it up properly, so it's not in the environment in the United States. Tetanus would be an impossible mission, so how do we protect ourselves? Because those spores are everywhere, y'all, in the soil. We can't get rid of it. Tetanus shots, right? You get a vaccine every 10 years. I checked. I did mine in 2008. I didn't even remember. Every 10 years to make antibodies against the toxin. Actually, you can get the bacterial infection, but it's an anaerobe. So unless you have a really bad wound, right, that's gone anaerobic, it shouldn't possibly grow. But if it does, it can still, even if you've been vaccinated, but you're going to be protected against the toxin it produces, and that's what hurts you, right? It interferes with your muscles' ability to relax. You stay contracted. So again, right, some comparisons. When we look at this, right, 25 degrees for our air sample and our desktop sample really lets us know probably the stuff on our desktop is coming from the air. Because this plate almost looks identical to this one, right? Our desktop at 37 looks a lot like all of our fingers grown at 25, right? That stuff, like we said, that grows best at 37 probably came from us, right? And temperature does affect growth. What grows and what doesn't grow. Look at the difference in these two plates, right? 37 versus 25, completely different, exact same source, right? Our desktop. But our air contaminants preferred the cooler temperature, and the stuff from our hands preferred that warmer body temperature. Right? So temperature really will affect organisms. And each organism, even ourselves, has a range of growth. We refer to it as cardinal temperatures. So each organism has the minimum temperature we have to have in order to grow. Right? You get too cold, you're either going to kill us or stop us. And stopping is typically what happens to bacteria. When you stick them in the freezer, y'all, you don't kill them. That's how we long-term store our bacteria is in the freezer. Right? You're not going to kill them. You're just going to stop them for a little while. When you take them out and thaw out that meat, they start growing again. Depending on how long you leave that meat out of the freezer will determine how many bacteria start growing again. Right? We refrigerate again to slow growth. It'll slow the growth rate. Right? So you can keep it longer before too many bacteria are there and you don't want to eat it anymore. Right? When the orange juice gets slid to the back of the refrigerator and you find it one day and it's cloudy, do you drink it? No, apple juice is supposed to be translucent, right? So if it's cloudy, what happened? A large number of microorganisms probably grew in there. Do you want to drink them? Probably not. Toss it. Right? Make sense? Milk comes contaminated, right? It's pasteurized, so hopefully most of the pathogens were eliminated. It still has microorganisms in it. They will grow over time. They will turn that milk into something you don't want to drink. We call that cheese. Right? Or other things, right, that you may not like. This is why your juices are pasteurized as well, right? So hopefully, but what happens the minute you open that container? You just exposed it to the air, right? You contaminated it. Stuff gets in. It starts growing, right? If you have someone in your household that drinks out of the community container, your saliva contaminates that container, right? When I see somebody do that, I'm like, that one's yours. I'm putting your name on it. I'm not sharing with you. It's a miracle I even kissed my husband. Not right now. He's sick. He's couching it. Right? My dog was not happy. She did not get couch time with Mommy last night because Daddy was on the couch. Daddy also didn't get his flu shot this year. Mommy and Dean did. He's miserable. Maximum. This is important because we can use this to what? What happens to growth? It stops, and it can even be, what could you do to an organism if you heat it too much? You decrease growth, you could also what? Kill it. Why do you think we cook our food? Why do you cook it to a particular temperature? Certain microorganisms that are found in chicken and beef, we know about what temperature will kill them. That's why they say cook to this temperature, all the way through to the core. Right? Not all organisms fit in the same grouping, right? So we are mesophiles, we're right in the middle, right? We like 37 degrees. You get too hot, you'll kill us. Some microorganisms will grow, right, at much higher temperatures. Oh, I don't have my temperature things, right? But psychophiles, right? Psycho actually means cold, and as far as I'm concerned, that cold is crazy, right? Okay, so we're talking about glacial lakes, right? Right above freezing. Psychotrophs, things that can grow in your refrigerator, right? These ones, could grow in us, like listeria, right? You got to watch out for those in your refrigerated foods. Mesophiles grow best at our temperature. These are all the pathogens that make us sick. Thermophiles, you're going to find these in compost heaps, Yellow Springs National Park in the hot springs, places like that. Not necessarily stuff that's going to make us sick. We're too cold for that, right? But they could produce stuff that could make us sick. Although I have to say I don't know of any. Do you, Andrew? I don't know any thermophiles that make us sick. No. And then hyperthermophiles, we're talking about deep ocean vents. I mean, these are things that are growing at nearly boiling temperature. They actually have found some archaea, which are microscopic organisms, actually quite not very closely related to bacteria at all, but they're also microscopic and single-celled organisms that can grow at boiling temperatures now. That's pretty crazy stuff. So, why do we make things sterile? The plates we use in lab will be autoclaved eventually to completely sterilize them. The measures taken to disinfect their tabletops are not that extreme. So there's a picture of an autoclave, right? You stick it in, we seal it up. It's a pressurized chamber. Steam, right, under pressure, penetrates through even the glass and everything. Will even kill endospores. It'll penetrate through that spore coat. It will kill any bacteria or viruses you stick in there. It will completely eliminate them, right? So we don't contaminate our environment, right? We kill everything before we throw away the Petri dishes and the tubes and all of that. Why don't we autoclave our desktop? Will it fit in there? No. That's one problem, right? We're not going to pick it up and go bring it over to the biology lab. It's not going to fit. Do we need that level of clean? No. Our Lysol is sufficient for what we're doing, right? We don't need it to be completely devoid of microorganisms. All right, so our microscopes. So hopefully you're familiar, but maybe it's been a while, right? If you need a little extra help, make time outside of class, come hang out with me. We'll have microscope fun. Next week you'll actually use them. So you have your ocular lenses, right, are the ones you look through. These are what you see the virtual image that was produced by the objectives. So it kind of looks like it's floating inside the microscope, right? The real image is produced by the oculars. There's several different ones that we can choose from that magnify different levels of magnification. The condenser is in your stage, right? That's the lens that's going to focus the light from the light source onto your slide. So when we figure out magnification, we're magnifying twice. The ocular lenses up here and the objective lens. So these are referred to as compound microscopes. So we have to multiply our magnifications. That image is only magnified once by the ocular. So even though you have two so you can look through, one of them is reflected, right? So that you can see out of both eyes. So like when I was in school, we had the ones with just one monocular, right? But even though you have two, you only are magnifying once by that apparatus, by the ocular eyepiece apparatus. So our oculars are all 10x. You'll see it stamped right on the lenses. And same thing with the objectives. The X is the times magnified. So all of them have their stamp on them. This is an example of magnification. I have to say I've never seen a 15x. I've seen a 20x ocular though. But these are kind of made up numbers just so you guys can practice multiplication. I do have calculators for you guys to use. Right, but you just multiply the two numbers. These are the actual numbers for our microscopes. So our microscopes, we have 4 lenses. The scanning lens, which has a red ring on it, is only 4 times magnified, times our ocular, gives you a total magnification of only 40. This is pretty much most of the time a complete waste of our time in this lab. It's just not enough magnification. Scanning means that you can usually see a very large area, right? It's not a very high magnification, so you can see a lot of the field of vision. We don't necessarily care so much about that in microbiology class because what we're looking at is teeny tiny. So this is a complete waste of your time once we start working with bacteria. Typically we start right here at low power, which is the yellow, which is 10x objective times the 10x ocular, which is only 100 times magnified. Again, not a whole lot of magnification, but is a good starting point. High dry although much higher magnification, this is your blue ringed one, 40x times 10 is 400. This one too isn't enough magnification for looking at bacteria. So we typically just skip over this one. The problem with this one too is that you're not supposed to get oil on this one. If you do, you're supposed to wipe it off. If you don't wipe it off this lens, it will creep in and make this lens permanently blurry. Most of the ones in this lab are permanently blurry. You will be wasting your time trying to focus at this magnification because students don't follow directions. It's true. Okay, not the case for you guys, right? If you accidentally get oil on it, you're going to wipe it off right away. When we get done with our microscopes, which we will be using oil all the time when we work with bacteria, you have to put the oil to be able to get the oil objective lens to come into focus, because you'll notice it's very close to the slide. And most of the light will get refracted as it moves through air because its speed changes. So in order to keep it smoothly moving through the lens, through the slide to the next lens, we put oil, which has the same refractive index as the slide, but oil, as you know, is movable. So that oil is going to fill the gap, and it's going to help bring that light in so you can see and focus. When you're done for the day, you take lens paper, and you wipe off until you don't see any more oily residue on your lens paper. And then you put your microscope away. You don't have to clean it in between slides, right? That'd be a waste of paper. You clean it when you're done. So that way when it's sitting, it's not creeping into the lens and making it blurry. That's 1,000 times magnified, and even then is not going to seem like a whole lot when it comes to bacteria. So the next thing we're concerned with as far as our microscopes isn't just magnification, but also resolution. How clear of an image, right? How far apart do those two little things need to be for us to see them as two separate little bacteria? That's determined by the ability of your lenses. So your lenses, in addition to a magnification, have a numerical aperture. This is the number assigned to the ability of that lens, including your condenser, 1.25 here, to be able to help focus that light. So I'm going to pick on you, Andrew. Do you need those to see me? Your glasses? No. So you need them for reading. Do you need yours to see me? Okay, all right. So I am going to pick on you. Okay, you ready? Take them off. How many fingers am I holding up? Don't tell her. 2. Okay, put your glasses back on. 3. Oh, good. Your prescription still works. Okay. All right. So what was the problem for her? If I'd gone like this, right, she probably would have seen it was 3, right? The fact that I had them all squished together made it impossible, right? She guesstimated, right? Because we use other things to figure out what we see, right? She kind of guesstimated it's definitely not 1, right? She probably could have guessed 3, but she was honest that it was 2 to her. They blurred together, right? She was trying to figure out by the width about how many fingers I was holding up. Does that make sense to you guys? So her lenses in her eyes cannot focus to the level that she needs to see distance. So she needs corrective lenses to focus at that length. So for our microscopes, they're set too. They have a set prescription. How good they can focus light. That's the number assigned to them. Just like her glasses have a number assigned to them. What she needs to correct for the inability of her natural lenses in her eyes. Make sense? Then as you age, the muscles get tired, right? And you have to start wearing reading glasses, which I don't wear. I just blow up everything on the computer. I don't read print text anymore.
Mom and Dad, they don't like the way I read.
I just forget.
They're like, oh, for me.
Yeah. I just forget. So resolution is this ability to clearly see and discern objects when they're very close together. We can actually mathematically calculate that, and you will have to do that in this lab. So again, I have calculators. We refer to this as the limit of resolution or the resolving power. The formula for it is the wavelength of light. The type of light that you're using really will affect the ability of being able to see and focus. So for our microscope, you'll notice they all have a filter on them because of that particular wavelength of light will give us the best resolution. Wavelengths, right, is measured in nanometers. So for this, it's going to give us a number. It's going to give us a length, right? Nanometers is a length. So we're going to get a distance, a length. We're going to know that number, how far apart, right? How far apart can I get them? And you're going to see them as separate. If I put them too close, right? If I'm not within that, if I'm not outside that limit, if I'm inside that limit, you're not going to see them, right? They're too close together. What's going to determine that? How good your lenses are. The condenser, which is focusing onto your slide, and that objective, which is creating that image for you. So we want wavelengths of light that are small, right? We want this number up here to be as small as we can and still see it, right? We've got to stay in the visible light spectrum. So for us, that would be blue light. So you'll notice the blue filters. It doesn't make what you see blue, right? It's helping us use that wavelength of light. And then, of course, we want the highest number for numerical aperture, which is going to give us the smaller number, right? So I don't know about you, but fractions sometimes freak me out. Okay. I call them evil Siamese twins, right? Because the top and the bottom are hooked together, and if you don't treat them the same, they get mad. In other words, you get the problem wrong, right? So 1/6, right? That's that piece of pie, say, right? Who wants the piece of pie? Who wants the rest of the pie? Yeah, you didn't eat breakfast either, right? Okay, so notice the number at the top is small here, small piece of the pie, right? Part over whole. What's left, big number, right? This is the bigger fraction, bigger number on top, right? Do we want that, though? Do we want a really big number or a really small number when it comes to resolution? We want small. We want to be able to put them right next to each other and we still see them as two separate things and they don't blur, right? So we want little. We want a little wavelength. We want big numerical apertures. Make sense? All right. So let's do, well, again, visible light, right? Blue is the best for us. Because we got to stay in that visible light spectrum. So let's do some math. So the filter they're using is 520. So where does that go? At the top, that's the wavelength. Where do we do with the 1.25 and the 0.25? We add them together at the bottom, right? There's our equation. You need to know and memorize and be able to rewrite this equation for next week. When we do out the math, this is our number in nanometers. This is a distance. 437 nanometers. That's how close we could put two objects and we will see them as separate. So, if we then put them 330 apart, will we see them as being separate? What's our limit? 347. Right? Are they within the limit? Yes. So will we see them? No. They are going to blur. They got to be outside the limit. The limit is minimum, right? The minimum distance. Are they the minimum distance? No, they're too close. Right? So they're going to blur. They got to be outside 347 to see them. So what if they are... What if we switch objective lenses and now we've got a better resolution, right? This is a smaller number, 247. So, of course, as we go up in magnification, so does our resolution. Otherwise, we wouldn't see crap. So, if they're 250, remember our resolution is 248. Will we see these guys? Yeah, they're on the edge, right? They're at 250, but they are at least outside 248. So we would see these two objects as two separate objects. under these conditions. So what's your microscopes? Our microscopes, again, we use a blue filter. It's about 500 nanometers. Our oil objective lens is 1.25. If we actually put oil on our condenser, which we don't bother doing, because that would be a big mess, we could have a theoretical best case scenario resolution of 200 nanometers, which is 0.2 micrometers, which is about the size of a bacteria. Which is why I said, even at 1,000, you're going to be wanting more magnification. So, with our eyes, we cannot see microscopic organisms, right? Because our resolution is only at about 100 micrometers. So, with our microscopes, we're 500 times better than our eyes. 100 micrometers is equivalent to 0.1 millimeters. So you see these little lines on here, on this metric meter? Those are millimeters. So those of us with really good eyes, you could envision 10 lines in between those little lines. Yeah, that's not me. Remember that whole problem with the reading? Some of you guys may be able to see to that level, right? But, you know, we do have about that level of ability with our eyes. The good news is in this class we're only measured down to millimeters, right? All of us can see those lines, right, on the ruler, I hope. Otherwise, get you some glasses. So this is a really cool website that I'm going to go to in a second, and that's, I think, our last thing. Remember, we started late. Be on time. Most of you guys were. I was just waiting for that other person, too. This is bacteria that are rod-shaped. Yep. That's about as detailed as we'll get under our microscopes. Here's some yeast. They're a little bit bigger. They're eukaryotic. We'll look at them next week. Here's some red blood cells and someone with a staph infection in their blood. These are actually bacteria and little clusters of tiny cocci bacteria. In lab, yeah, this is as exciting as it gets, except you'll color them, you'll make them pretty, you'll have fun with them. You're going to see rods, and you're going to see cocci, right? In different arrangements, like these are clustered up, these are forming chains. You'll see them all by themselves, right? The eukaryotes, which we're going to look at next week and that you're going to do for homework, those are the exciting ones. Although students don't usually look forward to them because you have to know all their names and identify them. All right. Oh, I do still have 2 more slides. Okay, so there's different types of microscopy out there. In this lab, we're going to use bright field microscopy, where the background is bright, right? Dark field is where the background is dark, and the specimens are illuminated. We could do this with our microscopes. We turn off the light from below and shine it at it from the side. But we don't bother with that. Mostly we just use bright field. We don't have phase contrast. I wish we did. That's the next grant I'm going to write. I'd like to get us some special filters that change the phases of the wavelengths of light, which helps give you better contrast. That's why it's called phase contrast. So notice this is bright field, this is dark field, this is phase contrast. You see how much more you see, right? You can actually see all the little guys outside of this euglena, right? Where they just look like dirt on these slides. These are actually little tiny organisms, right, outside of this euglena. Fluorescent microscopy, they actually might start doing across the hall in the biotech program, the science laboratory program. If you don't know about that new program we have at Delgado, come ask me about it if you're interested, if you love lab work. Some organisms are naturally fluorescent. If you shine UV light on them, they'll fluoresce. One of the organisms we work with, Pseudomonas, produces a green pigment that is fluorescent. If you shine UV light on it, it will glow blue. Some organisms, as I said, are fluorescent. We're going to make fluorescent E. coli towards the end of this semester. We're going to give it genetic information to make that protein and fluoresce under UV light. And then scientists use a lot of dyes nowadays that fluoresce, where they can label different parts of organisms and observe that under the microscope. And I have a beautiful poster in my office It shows all the different fluorescent dyes and different pictures that microbiologists have taken of labeled specimens with fluorescent dyes. This type of microscopy obviously uses UV light and special filters. You can't look at UV light, you'll damage your eyes. So the last thing I want to show you guys, and I have the link for you, or if I don't, I'll put it. Open. This is really cool. One of my students showed it to me years ago. It's an interactive website on cell size. And so you can compare, a coffee bean to a grain of rice, and you slide the bar. You can see amoeba, like we'll see next week, and paramecium, right? The egg is the largest cell in a woman's body. An ostrich egg is just one cell, right? Go down to the little sperm that carry the DNA. They're mobile DNA. They're important. They're tiny, but they're mobile DNA. We need it for the egg to make a being. Red blood cells. Then we finally get down to like yeast, poor little bacteria even smaller than the mitochondria in our cells, right? And then you got to zoom in even more, which, our bacteria are going to look about like that under the microscope. We don't have the ability or the resolution, right, with a light microscope to see viruses. You need something like an electron microscope, which apparently we're going to have here eventually too. I'm pretty excited. I thought Phil was joking, but really, apparently we're going to get one. And there are microscopes now, atomic fork microscopes that can go all the way down to single atoms, like the carbon atom. Oh yeah, because chromosomes are found in eukaryotic organisms, which are quite large.
But it puts DNA in there.
I'm trying to see where you are. Ribosome. Oh yeah, viruses are teeny tiny, y'all. This is a lot of DNA and protein compacted to form a chromosome. That's a lot more molecules than a virus. A lot. Especially if you're talking about a human chromosome. Did they label it? Yes, that's a human X chromosome probably. Lots of DNA and protein. Okay, so don't forget for homework, especially if you haven't used a microscope in a while, watch my video. I was going to play it in class, but we don't have time. Finish this homework packet if you didn't in class. Study and do your terms. Use that worksheet that I gave you as well. Do your quiz. Do your Lab 2 homework, which is reading exercise 3.3, filling in the blanks. Draw your organisms. Notice all these handouts are here in case you lose it. There's the textbook because you can't go buy it right now for Lab 2. Study, right? And so here's the lecture. Oh, I posted it twice now. I'll get rid of the old one. And then, what is this? Oh, that just takes you back to here. I'll post this recorded lecture as well. Okay? All right. See you guys next week. Yeah, don't keep the lab copies. We need them for everyone else who can't buy a book right now.